Transcription is the process by which we take a gene from the DNA and make a copy of it in mRNA using RNA polymerase. This process takes place in the nucleus, which you see here. Now, just like we saw with DNA replication, there's an enzyme called a helicase, which opens up the double-stranded DNA to expose the two strands of DNA here. Then another enzyme called RNA polymerase slides along one of the strands of DNA, and everywhere that it sees a DNA nucleotide, it puts a complementary RNA nucleotide directly opposite. Now, in DNA, cytosine and guanine were always paired together, were always opposite one another, and adenosine and thymine were always opposite one another. The same thing is true, it's still complementary base pairing, except that in RNA there is no thymine. Thymine is replaced by a nucleotide called uracil. So, when the RNA polymerase slides along the DNA strand, where it sees a C on DNA, it puts a G, an RNA G. Where it sees a G, it puts an RNA C. Where it sees a T, it puts an RNA A. And where it sees an A, it puts an RNA uracil. Okay? So the complementary base pairing is exactly the same, except wherever there would be a T, if this were DNA, we're now going to see a U instead. After the RNA polymerase finishes copying that gene, the mRNA is released from the DNA and is able to exit the nucleus to go out to a ribosome for translation.